Hello, everyone. I am Clare Gollnick. I am co-founder and CTO of Fathom Data. I'm here to talk to you today about designing data products for life and death decisions. Now, ultimately, this is a talk about our data infrastructure, but our data infrastructure is informed by our tech strategy, and our tech strategy is informed by our broader mission. And so in order to give you enough of the why as to why we made the architecture choices that we did, I'm going to have to start with the problem that we're trying to solve. Fathom is looking to simplify access to biomanufacturing data. Biomanufacturing data is process data. It is the records that are kept, the observations that are collected while a new drug is on the manufacturing floor. And this data is critical because it is the data that is used to answer the question, is this batch of drugs safe for release to patients? Now, if it sounds a little weird to use manufacturing data to answer a safety question, that's a little bit fair. But the perspective you have to realize is that for the vast majority of drugs that are manufactured on a day-to-day basis, they have already gone through a regulatory approval process. They have been approved and have gone through a clinical trial. So the question of safety is not as much one about newly discovering if the drug is safe, but about generalizing that inference, about saying, is this new batch of drugs that we just manufactured the same or similar enough to the batch of drugs that we manufactured during the clinical trial to say that if we released it to patients, it would be just as safe. So it becomes a question of equivalence. Are these two things the same? And that feels like a pretty simple question to answer, but it's actually incredibly complicated and unsolved. Yes, there are some cases in which after a batch of new drugs is manufactured, you can run a suite of tests, an assay, and you can say, yes, this drug product or this drug substance existed in this drug product at this concentration, at this specific purity. And if you're capable of doing that, then that means that your drug is likely a small molecule, like ibuprofen that's up on the screen right there. But if you have any of the more new or novel types of biologics that are now coming to the market, you no longer have a simple test that you can do after the fact. If you have a protein, for example, you can have identical chemical structures, but if the protein is not folded, it does not have the three-dimensional structure in the same way, it doesn't have the same bioactivity. Or if you're delivering a cell therapy, not only do we not have a functional assay to determine if two cells are the same, no one is asserting that any two cells are the same. Every two cells that come together are different. And so every time we deliver them to a patient, we sort of have to ask, are these functionally equivalent living organisms? And we don't have a definition for that. And even in cases where the active ingredient is pretty simple, perhaps like an RNA, you can sometimes run into problems with stability, where the half-life of the drug is so short and it degrades so quickly that in order to say, is this drug the same, is it active, you might have to push your quality decisions all the way out onto the edge, past the distribution and away from the manufacturing floor. This is a really complicated problem. The industry knows about it. This is not news to anyone working in biopharma, and they have a solution to it, and their solution is to say, the process is the product. As long as I manufacture things in exactly the same way, then I can be sure that I'm delivering the same product. And the way that I convince myself, my patients, and the FDA that I'm delivering this safe and effective product is to prove that I have control over the process. And so this process data, the manufacturing data, is critical for making these batch release decisions that determine whether a drug is safe for patients. Now, Fathom's a relatively new company, and we're really interested in this process data. And one of the reasons you might imagine that we are interested in it is because we are planning on automating this batch release decision. That we are going to say, okay, we've collected all the data, and now we know objectively that this batch of drugs is safe for release. But Fathom is not trying to automate that decision. And one of the major reasons we're not trying to automate that decision is because we fundamentally don't believe that data is objective. Now, or at least data is not inherently objective. It's not innately objective. And sure, sometimes data can be objective, but it has to meet certain conditions. And one of those conditions that must be met in order for data to be objective is that all stakeholders must agree on the objective. And for however hard all of us in this room know it is to keep a data set up to date, trying to keep a large group of people aligned, that's a whole different story. But again, the question we're talking about right here is we're trying to deliver safe and effective drugs to patients. So how is it possible that we have a stakeholder that doesn't agree with this objective? Like, wouldn't it just be obvious that everyone wants to do this? Well, this is where it gets tricky. Because an objective is really easy to get alignment on if you only say the rosy part, if you only say the easy part of the objective. We're all trying to save lives. Everyone's going to agree to that. But what is hard about making a stable and reliable and robust decision system is gaining alignment on what you do in a marginalized use case or a marginalized decision. And that's because the stakeholders often don't agree on the cost of a failure. And in medicine, this arises every day, all the time. There's often this tension between individual ideal outcomes and what is good for the collective, or maintaining trust, public trust in our system of medicine. And this happens all the time, but in a specific batch release decision, this might happen if a new batch of, let's say, a cell therapy is being manufactured, and during that, something goes wrong with the process. Let's say it gets contaminated, some bacteria grow in with the cells are not as healthy as they needed to be or they would have been if they'd been used as part of a clinical trial. Do you release this batch? Most regulatory agencies looking out for the collective good are going to say no, because I can't let a potentially contaminated batch of drugs out into the public. But right at that moment, there are patients who are sick, who are suffering, and who are potentially terminally ill, who potentially by the time a new batch is manufactured will no longer be treatable. Perhaps their disease has progressed so far that there's no recovery. They value their time so much more and so much differently than the people who are making a decision for the collective good. And so this is an ethical decision, and it's not a place for Fathom to operate. So what are we doing instead? We believe that data is a tool that different stakeholders can use to meet different objectives. And so we clean data, and we do QA on data. We make sure our product is great so that our customers have an easier time making their own quality decisions for their own products. And so we write a lot of data cleaning algorithms. But when we write those data cleaning algorithms, we are taking the lessons we learned about what it means to use data objectively. And that key lesson is that how your algorithm fails and what happens when it fails is the very first thing you should think about before an algorithm goes into a system. And we put constraints and requirements on any algorithm that goes into our system. And one of the constraints that we put on it, which is a little bit counterintuitive, is that we ask our algorithms to fail. Sometimes to fail visibly, loudly, but sometimes to fail intentionally. In a situation where they maybe could make a good guess, and maybe that guess would even be right 90% of the time, but that extra 10% of the time, the consequences of that mistake would be too high to be worth putting out an answer. So we make our algorithms a little less good than they potentially could be based on the tech or based on the data, but we do that strategically because we understand the cost of failure in our use case. But what's cool about this is that when you assume failure or even encourage failure, you get some really cool data architectures coming out. And our system has to be designed from day one to be robust to our algorithms or systems and our data pipelines failing. And so that means we can't at any point put anything where there's a single point of failure or where there would be a cascading failure. And instead, our entire system is designed with distributed, redundant, sometimes, data pipelines that are all asking a small part of the question and voting on their own specific small part of the problem. And this actually creates a lot of engineering headache on like the first 2 weeks this system is deployed. But longer term, it creates a much more robust system because everything is engineered to not assume anything else worked and to make its best guess given the information that is available at the time when it needs to make a decision. Now, what you might see on the end there is a big black box that looks like a single point of failure. But even in our data store, we're actually, in order to maintain auditability, we're actually maintaining separate layers of our data. Now, it's pretty hard to get an intuition really fast on what biological layers might look like. So instead, to get an idea of what a layered model is, a really commonly understood layered model is that of a map, where you potentially have land masses and water masses at the bottom, and then cities and countries and buildings and traffic, and each of these different layers, they have different levels of reliability, right? Like, yes, technically the continents are moving, but I probably don't have to include that in my model because it'll be good enough, right? So there's some level of base, high-performing algorithms, and then you can get your more and more robust, potentially more error-prone algorithms on the top. And because we don't combine these layers when we store it, we get to combine them at query time. And what's cool about that is at the time in which we query our data, we know who is querying our data, which means that we can dynamically construct the world as they view it. There isn't a single source of truth because everyone looks at the world differently. And they're going to look at different things and want a different representation of that data. So this has been a little abstract. So we're going to go very concrete into the world of what actual biomanufacturing data looks like. And maybe you might be surprised to know that it looks like a piece of paper, because that's where pharma is right now. Almost all their data is stored on paper. And so when we get a new piece of paper into our system to analyze, we are deconstructing it, just like you saw, like we talked about, and we are analyzing this piece of paper, and we might analyze it in many different layers and look at it from many different perspectives in order to make inferences about how we should clean, harmonize, standardize this data. For example, if you get rid of all the words and you just look at the lines, you'll notice that a lot of relationships are actually maintained and information about relationships between different data. So even when you don't know what the data is at all, you know that a relationship in the same column is related to another piece of data that is in the same column. And so you can start getting this idea of how your data and how your schema is represented, even without having any access to the text at all. And then perhaps you can look at the layer with the text alone, and you can make different inferences. You can infer and look and identify where all the dates are and ask yourself, what can I learn about that? Or you might throw an NLP model on there, do some content analysis in order to group and organize data. You can do all of these different things, but they're all done in parallel and separate from each other. We are not trying to push them all into one giant model in order to understand this data set. So what does it look like to dynamically reconstruct different people's worldview at query time? Moving way down and away from the life and death decision of if a batch should be released into like one of those more practical work issues that teams in biooperations tend to have. You have two teams on a traditional operations team. One might be the manufacturing science team, and the other one is the quality team, and they have different goals. Right? They have different objectives. The manufacturing science team is going to look at how their process is doing over time. They want to see, if I started incubating my cells at a slightly different temperature, or if I used a different pH buffer, what did it matter? And so they're going to be really looking at the content of the data. While the quality team needs to ask each individual batch, did it happen exactly the way that the process had aligned? And so they're going to looking for any specific anomaly or deviation on that batch. So if you look at something simple like this crossout right here, to the manufacturing team, they're only going to be interested in the corrected version of this data, the one where someone crossed it out and then wrote something on top. To them, that is the only piece of data that matters. The other one might not exist, right? And so you want to be able to represent that worldview to them when they ask for the data. You want to be able to say, yes, this is what this number was on this batch. This is what the cell viability was. But the quality team is going to be able to want to ask, hey, how many times did somebody cross this number out? Because for a couple of reasons, we might be looking for fraud, we may be needing to retrain our team, we might be needing to do a lot of different inferences to make sure that if this resulted in a deviation, that it was handled and documented appropriately for the FDA. And so because we maintain and keep and model our data as all these separate different pieces, we can represent these different worldviews on the same set of data. So in closing, a couple ways of how Fathom's perspective on data and the fact that data is not objective inherently in and of itself plays into how we build our data teams and data architectures. The first is that we believe that data is a tool, and that we can use that tool to do many different things. But that does not mean we know everything, right? We're very careful about where we should be making decisions and where we should not. We believe that algorithms always make mistakes no matter what we do. And so sometimes we should make them make mistakes. And that the broader ecosystem in which you introduce your product to these customers, they should be aware that your algorithm should make mistakes too, and it should be exposed to them so that they can correct it, and it becomes part of their understanding and their interaction with the product as well. And then understanding that what happens when your algorithm fails is probably one of the most important assumptions you can discuss before you even start building something. It is incredibly powerful. And then the last one is sort of just a small thing, but we talk a lot about single sources of truth. And I really think we should start thinking about being open to keeping multiple copies of the same data, because there isn't only one way to look at the world. And redundancy is a critical part of how you build resilient and robust systems. And so I think we should look into that a little more. And if any of this is interesting to you, and this is how you like to think about data, you should come talk to me, because we're looking for some great people for our team. So thank you.